have the most threats. So that's where we started. So we started actually quite broad, so national level, and then in the end,
Number one we are going to discuss is leaf spot. Leaf spot is a disease in which there is formation of several spots over a surface of leaf, such that these leaves spots are caused due to the over deposition of anthocyanin. Due to the over deposition of anthocyanin, there are formation of several leaf spores. Over deposition of anthocyanin, formation of these spores. After which these leaf spores are formed due to over deposition of anthocyanin, and apart from that, apart from that, when it comes to the agent, causative agent which is responsible for in this case is basically what we consider as the xanthomonas. Xanthomonas is responsible for it. Xanthomonas campestris. Xanthomonas campestris is responsible for the leaf spot. Xanthomonas campestris. Let me just write it there as well. Xanthomonas campestris. And here, the condition which it causes is unfavorable, because here it's due to the over deposition of anthocyanin, the process of photosynthesis is unable to occur properly, due to which the growth of the plant is ultimately diseased, and as a result of that the plant is unable to grow, uh, because there is effect on the leaves of the plant, due to which there is no proper food synthesis, due to which there is no proper metabolic activity occurring within the plant. And when it comes to its prevention of for this includes several methodologies, which include, among which there is hoeing, which means removing the plant physically, removing the affected plant physical removal of the plant, as well as hoeing, cutting of the plant which is infected, as well as we can utilize several uh, pesticides in this case. Moreover, there are certain other strategies as well, such that we can also uh, grow the plant. We can also utilize a methodology of crop rotation, in which we are going to rotate the crop, such that if we are having one crop grown in one season, let's suppose if we talk about rice. So rice is a crop which is grown in summer. Let's suppose it is grown in summer. So after summer, when it comes to the winter season, in winter season we are having a season of wheat, and after winter, when we are again having summer, we should not grow rice this time, but rather we should grow another crop which is similar to rice. This methodology is known as crop rotation, and crop rotation can be utilized in order to prevent such conditions. And apart from that, there are some other strategies, such that we can utilize seeds which are well screened, because the disease leaf spot is a seed borne disease. It is a seed borne disease, which means it is born through the seeds carried through the seeds, due to which we should utilize screened seeds which are without any sort of pathogen in it. Apart from which, when it comes to the second part which we are having, which is about the blight. What is blight? Blight basically refers to the formation of dark blights over the plant. Now, for this, as you can see in the diagram, there these are examples of how the blights are formed. As you can see over the leaf, these are the formation of several blights. And uh, which organism is responsible for this? Uh, the organism which is responsible in this case would be Xanthomonas oryzae, for which causes blight in rice. And when it comes to the leaf spores, Leaf sport mostly occurs on plant species of Solanaceous family. Plant species of Solanaceous family. Leaf sport mostly occurs on the plants of Solanaceous family. Apart from which, when it comes to the blight basically mostly occurs in the Poaceous family. Mostly occurs in Poaceous family. These are those plants of rice, as well as crops of rice, wheat, and all that, which are not in the Poaceous family, family Poaceae. And when it comes to the organism, it is Xanthomonas oryzae, which affects the rice, as well as there is Erwinia amylovora, which cause the blight disease. And apart from that, when it comes to the prevention is same, that we are going to utilize some pesticides, or we are going to cut the crop, or utilize tested seeds for this, because this is also a seed borne disease. Apart from which, when it comes to the second part which we are going to discuss about it, is about the bacterial soft rot. Bacterial soft rot is a condition in which the bacteria in which the plants in which the fruits of the plants get soft rotted, such that their the cells of the plant tissues become turgid, as a result of which they tend to as a result of the necrosis factor, as a result of necrosis occurring within the cells, the cytoplasm comes out, shrinks outside of the plant tissue, as a result of which the plant tissues become dead, appear black, and seem watery due to the release of cytoplasm from the dead plant tissues. As a result of which, this condition occurs, which leads to the formation of bacterial soft rot. And when it comes to the bacterial soft rot, there is organism known as Corynebacterium. Corynebacterium is responsible to cause air rot of wheat, and we are also having example of Armenia Keratovora, which is responsible for it. Armenia Keratovora is responsible for it. As you can see in the diagram, these are the soft rods occurring. And prevention of this, again, crop rotation, utilization of screened seeds. And when it comes to the soft rot is again a seed-borne disease. Seed-borne disease, actually. And apart from that, when it comes to another disease which we are going to discuss over here, is also considered as a cancer of plants in plants. And that cancer in plants is considered as bacterial gall. This bacterial gall occurring in plants of particularly, it basically occurs in woody and herbaceous plants. Woody and herbaceous plants which are having, for example, trees, or those plants which are having herbs and treated are infected by bacterial gall. Now, bacterial gall basically is caused by two major organisms as per our book, which includes Rhizobium leguminosarum, which basically causes to infect uh, the bacteria, also infect the plant by formation of small outgrowths on the lateral sides of the plants. Several outgrowths on the lateral sides of the plant. As you can see, several outgrowths are formed, small outgrowths, abnormal outgrowths of cells are grown, which is considered as a tumor. So here it is grown due to mutation occurring, as a result of which we can consider Rhizobium leguminosarum as a mutagen, a biological mutagen. Apart from which, this outgrowth occurring is what we consider as the gall, which is the abnormal growth of cells. What we consider as gall in the abnormal growth of cells. In case of leguminosarum, Rhizobium leguminosarum, the growth occurs in legumes, particularly in the root nodules of the, particularly in the root nodules of the bacteria, root nodules of this, plant, in the legumes, due to which this is considered as root nodulus bacteria. Apart from which, when it comes to the gall, apart from which when it comes to the gall, Another organism which causes this includes Agrobacterium tubificium. Agrobacterium tubificium is basically a bacteria which involves a plasmid, a special plasmid which is having the, the genes for tumor formation, and that genes for tumor formation are contained in its plasmid, which is also known as tDNA. tDNA, and that tDNA has a capability of formation of tumor, such that whenever the Agrobacterium tumefaciens infects Agrobacterium tumefaciens infects the plant, it causes to insert its tDNA into the plant genome. As a result of which, the plant genome being integrated with the tDNA causes to now become mutated, which means now it has a gene to form abnormal growth of cells of the tumor or the gall in case of plants, which will further be grown out as the bacterial gall, as a result of which this tDNA is responsible for it. Moreover, the Agrobacterium tumefacium is also utilized a lot in genetic research or genetic engineering, by in which the main agent which is utilized for from the Agrobacterium tumefacium is again the tDNA, but this time whenever we utilize it in genetic engineering, what we actually do is that utilize the enzyme restriction endonuclease to cut off the tumor causing part, cut off the disease causing part or tumor causing part of the Agrobacterium tumefaciums, and instead of the tumor causing part or disease causing part, we add into it the gene of another bacterium such as Escherichia coli, in order to synthesize, let's suppose, a vitamin K or vitamin B12, sorry, vitamin K and vitamin B2, as well as for the synthesis of any hormone such as insulin. We add into it, after which, when we add the gene for the synthesis of the particular type of protein we want, we just add we just add into it and integrate into it using the enzyme which is uh, utilized over here, known as DNA polymerase. After the utilization of DNA polymerase, it causes to give us a modified organism, genetically modified organism, GMO, and that is now utilized for the formation of the desired product or the formation of the product of interest. So this is how Agrobacterium tumefacium is utilized. But on the other hand side, it also causes a disease which is known as gall, and it is type of cancer in plants. When it comes to prevention, prevention includes crop rotation, removal of infected plants, tested seeds, as well as it is a soil-borne disease. It is a soil-borne disease such that it is carried through soil, due to which removal of the infected plant is mandated in this case, as well as removal of the plant is mandated in this case. So this is about it. And when it comes to and the last disease which we are going to study over here is about the bacterial wilt. Bacterial wilt is basically a disease which is caused by the Pseudomonas solanoceram. In this disease, what actually happens is that there is loss and turgidity of the bacterial blades, bacterial leaf blade, loss and turgidity of the leaf blades, as a result of which the plant loses its turgidity. Moreover, the causing agent Pseudomonas solanoceram has a characteristic that it basically causes to block the xylem vessels of the plant, as a result of which the water is unable to be transported to the upper parts of the plant, due to which the upper parts of the plant become wilted, as a result of which this results into a condition of wilting, due to no water being reached to the upper parts of the plant, as a result of which there is no process of photosynthesis possible, as a result of which there is no glucose synthesis, due to which there is no metabolic activity uh, possible in upper parts, due to which there is wilting of plants in the upper parts, due to which this condition is known as bacterial wilt. And here two organisms are noted: Pseudomonas solenoceram and Arvenia trachyphylla. Arvenia trachyphylla and Pseudomonas solenoceram. When it comes to these organisms, they block the xylem vessels, as well as these are seed-borne diseases. Apart from which, when it comes to example of how to prevent it, prevention of this basically again occurs through the methodology of utilizing sort of pesticides, as well as removing the infected crop rotation, as well as utilizing of tested seeds. And apart from that, these are the methodologies. And moreover, this is a disease which mostly affects the solanaceous family. So solanaceous family, which includes tomato, potato, as well as we are having tomato, potato, onion. All of these which are involved in solanaceous family are affected. Apart from which, this was all about this video. And one more thing I would like to mention over here is about the leaf spot. Leaf spot is caused in tomato and potato. It is caused in tomato and what we consider as brinjal as well. So tomato and eggplant. So tomato and eggplant has the disease of leaf spot occurring in it. So this is a point to be considered, and not in solanaceous family, in the leaf spot. And when it comes to the bacterial blight, it is caused in poaceous family. So this is all about this video. Hopefully we'll meet again in another video.